Hi class, in this recording we are going to be focusing on the cells of our immune system, particularly the innate immune system. So let's dive right into it. As we look at these cell types, they are going to include lots of phagocytes. These phagocytes are cells that engulf foreign matter, solid foreign matter. And to review, phago means to eat, cyte means to means cell. And we also are going to have a lot of leukocytes. The leukocytes included are the neutrophils, eosinophils, basophils, monocytes, and lymphocytes. These leukocytes are the 5 kinds of white blood cells that we talked about when we covered our unit on the circulatory system. So let's dive in and talk about neutrophils. Neutrophils are the most common kind of white blood cell, and they're going to be wandering through the connective tissues of our bodies, destroying bacteria. Neutrophils are really good at destroying bacteria more than any other function. So think of these neutrophils as bacteria-destroying white blood cells. And they can destroy bacteria via phagocytosis, and after they phagocytize the bacterium, they digest them. Our neutrophils can also destroy bacteria by committing suicide or self-destruct. You can think of them as kind of the kamikaze soldiers of the immune system. And I say that because they can release a cloud of bactericidal chemicals. These bactericidal chemicals are going to be stored in lysosomes, and as those lysosomes degranulate, they are going to release their highly oxidative enzymes and chemicals into the extracellular environment, causing a respiratory burst. And this is called a respiratory burst because we have lots of waste products from cellular respiration bursting into the external environment. These waste products include superoxide and peroxides and hypochlorous ions. You could think of this as superoxide we don't have a common analogy to in everyday life. Peroxide, you can think of hydrogen peroxide, and when you think of the hypochlorous ion, I want you to think of the bleach. So we are chemically releasing hydrogen peroxide and bleach to the extracellular environment during this respiratory burst. And we find that around the outside of the neutrophil, we have a killing zone. All the cells around the outside of that neutrophil are going to be hurt. They'll be harmed by that respiratory burst. It destroys several bacteria. The problem with this is that respiratory burst also destroys the neutrophil itself. As we look at eosinophils, our eosinophils guard against parasites. So we have high concentrations of them in our mucous membranes, where parasites are typically going to be gaining access to our bodies. Our eosinophils also are going to monitor allergic responses and defend against a wide variety of other pathogens, but primarily, this is really what makes them unique. They help to defend against parasites. So if we have a multicellular parasitic infection, such as tapeworm or roundworms, it's our eosinophils that will help to fight them off. As we look at an eosinophil, to review, eosinophils are a granulocyte, and they have lots of pink-colored or eosin-colored granules within their cytoplasm. Eosinophils are also capable of releasing superoxide, hydrogen peroxide as well, just like the neutrophils do, but the eosinophils don't do it to such a degree that they destroy themselves in the process. Eosinophils are also going to help to activate basophils and mast cells within our bodies. And as we have those basophils and mast cells activated, that helps to moderate and control the inflammatory response, or what we typically think of as the allergic response. Our eosinophils are also going to phagocytize any antigen-antibody complexes. So if we stick and cover something with antibodies, our eosinophils are going to be capable of phagocytizing anything that's covered with our own antibodies. We should also emphasize that eosinophils will limit or downregulate the action of histamine and other inflammatory chemicals. They help to give us balance or maintain homeostasis so that we don't have that runaway inflammatory response or allergic reaction. Up next we have a neutrophil. a basophil, B for basophil. And as we look at the basophil, this is the last granule site we're going to talk about. As we look at the basophil, it's going to be filled with granules that stain very dark purple, crystal violet. And these basophils are going to secrete chemicals that aid in the mobility and action of other leukocytes. They're going to help with the inflammatory response and vasodilation. Basophils also will secrete leukotrienes. A leukotriene is going to activate neutrophils and eosinophils and attract them or draw them towards the site of infection. Basophils release histamine, which acts as a vasodilator, causing blood vessels to enlarge in diameter, which increases blood flow. And if you have faster blood flow, the leukocytes can get to the target area faster. Basophils also secrete heparin, which inhibits clot formation. We don't want blood clots because those blood clots would make it difficult for other white blood cells to travel through the bloodstream to get to the site of infection. Our basophils are intricately linked to mast cells. You can think of a mast cell as an activated basophil or a subcell category underneath basophil. And mast cells also secrete these substances. Mast cells are going to be primarily located within connective tissues as a connective tissue cell type, and they are very similar to basophils. We also have lymphocytes. This is an agranulocyte, and there's the most variety within our lymphocytes compared to any other category of cells. Our lymphocytes can be broken up into three broad categories: T lymphocytes, B lymphocytes, and natural killer cells. Of all the cells of the immune system, natural killer cells are probably my favorite. Now, as we look at our lymphocytes, most of our lymphocytes are going to be T cells, followed up by B cells with a very small proportion of natural killer cells. And they do many different things. There are very diverse functions for our lymphocytes. Natural killer cells are part of an innate immune response. And then as we look at the adaptive immune response, we have the T cells, the B cells, and the helper T cells that are going to function, the T and B cells that function in the adaptive immune system, and then the helper T cells that function in both the adaptive and the non-specific or innate immune system. So the helper T cell, which we haven't talked about in a lot of detail, is a crossover cell type. It's both part of the non-specific immune system and the specific immune system, or the non-adaptive and the adaptive immune systems. So let's talk about natural killer cells in more detail. You may have noticed I skipped over T and B cells. We're going to have separate lecture recordings dedicated to those cell types because, oh man, there's a lot of stuff to say about T and B cells. Let's talk about natural killer cells, though. Natural killer cells are a kind of lymphocyte that's going to be patrolling the body continuously, looking for either pathogens or diseased human cells. When we say host cells, these are human cells that have an intracellular pathogen. Natural killer cells are going to destroy any bacteria that they run into, any transplanted cells. So if we have a tissue transplant and we don't have good donor to recipient matching for that organ or tissue transplant, then the recipient's natural killer cells have a habit of destroying the donated tissues. Our natural killer cells are also going to destroy cells that are infected with viruses, and they will also destroy precancerous and cancerous cells. Our natural killer cells will recognize any cell type that doesn't belong into our bodies and attach to it or bind to it. After it binds to it, that natural killer cell releases a bunch of proteins known as perforins. So here's some foreign cell that we don't want in our body. The natural killer cell right here is bound to it. It releases the perforins. The perforins make a pore in the outside of the cell, and after the natural killer cell punches a hole in the outside of the cell, the natural killer cell is then going to release granulozymes that are going to be drawn into the target cell, and those granulozymes are protein-degrading enzymes. So the natural killer cell first perforates its target cell, and then releases digestive proteins to digest the target cell from the inside out, and that is going to make the target cell have apoptosis induced. so that the inside of the cell is destroyed and the cell self-destructs. And then afterwards, there's leftover cellular debris. That leftover cellular debris from the cell the natural killer cell has destroyed is typically going to be phagocytized by the macrophage and then chopped up into bits and shown to the adaptive immune system, because our macrophage is one of multiple antigen-presenting cells that we have within our bodies. Speaking of monocytes and macrophages, monocytes are the largest white blood cell within our bodies, and they typically are going to go from the bloodstream to connective tissues. Once they get into the connective tissue, they transform into the big eater, the macrophage. And as we look at the macrophage, we have two different categories. We have fixed and wandering. Wandering macrophages are constantly moving around, actively seeking out pathogens. They are very widely distributed, particularly within the loose connective tissues of our bodies. Think of areolar connective tissue. That's the one that pops into my mind almost immediately. My favorite example of a wandering macrophage is from that 1950s B-movie classic, The Blob, which is a large single cellular organism that starts to attack and swallow an entire town. So if you're looking for a fun movie to watch, one of those movies where you can have some popcorn and make fun of how cheesy it is, I highly recommend The Blob. It's hilarious. As we look at our fixed macrophages are macrophages that are stuck in a specific tissue. They can only work in the specific area that they have matured in. So as we look at microglia are one of the neuroglia of the nervous system. They are the macrophage of the central nervous system, or the primary white blood cell of the central nervous system. We have alveolar macrophages that are going to be located within the alveoli of our lungs to phagocytize any dust particles that get into the alveoli. And we also have hepatic macrophages. And I love this new name, hepatic macrophages. It's a very clear, concise, you know exactly what it means in terms of that name telling you what the cell does and where the cell is located. The old school name, the crusty name for hepatic macrophage is Knufer cells. So if you ever run into that, this is going to be in older literature. If you run into the phrase Knufer cells, K-N-U-P-P-F-E-R cells, don't quote me on the spelling of that. I'm terrible at spelling. The Knufer cells, or hepatic macrophages, are macrophages of the liver, and they stay in the liver. That's all we have for our discussion of cells of the innate immune system. If you have any questions or comments, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.